In this video, we're going to have a look at prokaryotic and eukaryotic DNA. We're going to compare their structure, how they are organized within the cell, where they're found within the cell, and how the genes are structured in these two different types of classification of organism. So when we start off by looking at DNA, DNA and RNA are the same for both prokaryotes and eukaryotes. In other words, they're made up of the same bases, A's, T's, C's, and G's in the DNA. They are both double helixes, and the RNA that is used to turn the message of DNA into proteins is the same. It has 4 bases as well. U's for uracil, C, A, and G. And the code for actually making proteins is also the same. So the code in the mRNA that gets turned into amino acids or translated into amino acids, the code is the same. It's universal, regardless of whether it's bacteria with prokaryotic DNA or it's a fungus or a human cell or a plant cell. The code is the same. But there are fundamental differences within the cells as to how these are organized and where they're found. So in prokaryotic cells, which is all bacteria, remember that prokaryote means without a nucleus, you can't have DNA inside a nucleus because there isn't one. But thankfully, prokaryotic DNA is a much smaller entity. It's a much smaller genome. And you can see in this diagram that it's organized into a circular chromosome and just one large circular chromosome. Therefore, there's no ends. It's just one big loop. And there are additional plasmids. You can see those over here. So we've got one circular chromosome that's here, circular chromosome, and additional plasmids. And the plasmids can hold interesting features like antibiotic resistance, for example. And if we have a look and contrast this with eukaryotic DNA, remember that eukaryotic cells include fungi cells, animal cells, plant cells. The place where we'd find the DNA, obviously, is in the nucleus. But maybe less obviously, although you might remember it, there's also DNA found inside a chloroplast and inside mitochondria. So we'll come to their organization in a moment, but we'll start off by having a look at the organization of the nuclear DNA. So you remember the DNA is arranged in chromosomes. There is lots of DNA inside a eukaryotic cell. In a human, it amounts to about 2.2 meters of DNA in every single cell. So we have to organize this carefully. So we put it inside a nucleus. The nucleus has a double membrane. It's organized in linear chromosomes. So that means that they have ends. And the ends are called telomeres. And telomeres might crop up when we talk about disease, because as telomeres shorten, you could actually end up with some diseases they think are linked with that. Now, when we have a look at this part of the diagram here, we've got the organization of the thin strand of double helix of DNA around proteins. They're called histone proteins, and it's like having the DNA wrapped like a piece of cotton around a cotton reel. And these proteins are really important. So here they are, they're disc-shaped. They're really important for wrapping the DNA. It actually kind of goes around twice, like sort of 2 wraps, and then it goes on to the next histone. And that's really important because it organizes the DNA so that when the chromosome condenses during cell division, it doesn't just go into one big tangled mess, but also it locks away the inactive parts of the chromosome. And as we've already said, there is lots of DNA inside a eukaryotic cell, so we have to be able to organize it so we can activate genes that we want turned on at the right times, and if you want them turned off, we can wrap them around the histones and lock them away. And we'll think about the implications of that later on in this course. Let's contrast that with prokaryotic so we've looked at those two organizations, so the circular DNA, the linear DNA, but now we'll also look at what actually the genes are structured like. So in bacteria and other prokaryotic cells, you have this limited amount of genome. So you've got to get all of your genes into the chromosome, and you haven't actually got any room for junk. So what we find is that you have in prokaryotic genes that overlap with each other. There is little or no junk DNA, and junk isn't really the right word for it because it usually would have a function, but in prokaryotic genes, there is no or almost no non-coding DNA. So all the DNA has a function, and you actually group similar genes together if they're going to have a similar job. So you can see in this picture we have what's called an operon, and an operon is a group of genes. So you can see 4 genes here, A, B, C, and D, and they are all going to be controlled by one promoter region. So here is our promoter. You can show that actually with a little arrow like that, saying join onto the DNA here, and let's copy the information that we need from these 4 genes, and we copy it all at the same time, and we make 4 proteins from it. So if all these were involved in the same metabolic process, maybe they were metabolizing lactose sugar, and you need all these 4 proteins to do that job, they would all be turned on at the same time and all made at the same time. Now, interestingly, mitochondrial and chloroplast DNA look very similar in their organization to prokaryotic DNA. And that's probably because the theory goes that mitochondria and chloroplasts were once free living prokaryotic organisms. Now, let's contrast this very tight structure of all the genes being close together, organized in functional groups, with what we see when we have a look at the genes in eukaryotic organisms. So here is our gene structure in a eukaryotic organism. Looks a little bit more complex. We have blue areas, red areas, and what we actually have here is coding DNA and non-coding DNA. So I didn't have a red post-it note, but we've got a pink one. So the coding DNA is the red bits, and they are called exons. So we've got exon 1, exon 2, and exon 3. These are going to end up in the final protein. But we've got junk. Well, it's not really junk DNA. Remember, I said it usually has a function, but we have junk DNA, bits that don't code for the final protein, in amongst the gene. So we're wasting space. But we can do that because eukaryotes have lots of space. So in between, we have these things called introns, and the introns are not going to end up in the final protein. So we've got 2 introns here. So it's interesting because they're using up space, but they have that space to use, but it's a different structure. So we've got much more spaced out. You can actually have genes that are related to each other on completely different chromosomes, spaced out all around the genome, and there's lots of non-coding DNA in eukaryotic genomes. So when we are looking at comparing prokaryotes and eukaryotes, there were lots of similarities at the beginning in terms of the actual molecules, the DNA and the RNA and the codes, but there are also quite a lot of differences.